the symposium is to maybe discuss a little bit some of the challenges that we will have in translating some of this preclinical research that we do in the GI disease arena into making viable drugs. Many of these challenges are across the board for all sorts of diseases, but I think there are some specific ones that we're faced with if we want to try and treat GI disorders. So this is one of the generic ones, and I think most of you will be aware of this, is the long time that's required to make a new drug and the massive attrition that we're faced in the different phases of the drug development process. We start off screening many, many hundreds of thousands of compounds if we're going for low molecular weights. We'll get some that will hitting our target, we'll test those in various in vitro assays, and we'll end up with a few that we will hopefully take into the clinic in phase 1 testing. And after several years, we will, with a lot of luck along the way and obviously hard work, end up with one new medicine. And this process actually has not got any shorter over several decades. Clearly, there was a lot of hope with the development and impact of new technologies. All of these have come in play in the last, say, 15 to 20 years. And they clearly have had an enormous effect on the drug discovery process. In particular, combinatorial chemistry, the high-throughput screening and high-content screening, and of course the advent of biologicals as therapeutics. But if we look to see the impact of these, they hasn't really translated yet into new drugs. We can argue, of course, that sequencing the genome, there hasn't been enough time yet to translate that into new drugs. But I think we could argue it hasn't really given us many more new targets that we could really hang our hat on and say this is a new target rather than just a gene association with a disease. High-throughput screening has massively affected what we can do and how many compounds we can screen at our targets. And pharma companies tend to get a little bit macho about how many compounds they have in their library. But we all screen well over 1 million very, very regularly each year. But as you can see, even this hasn't translated into an increase in a number of NMEs, so essentially new drugs. And what's even more scary is if you look at the number of new drugs that actually are based around new targets, and that's actually got worse. And I think this is actually pretty scary. So we need to somehow improve, not just through technologies, but I'd argue choose what we're working on and what targets we're going to work on. So the GI disease arena. Brendan discussed most of this morning, and I'm rather pleased that I think the diseases and the unmet needs that I've put up here and I will put up here essentially agree with him. If you look, there are different types of disorders. We've got the motility disorders or the functional disorders near the top, and you can see that the prevalence of these is absolutely vast. And then down the bottom, we have the more inflammatory disorders, and the numbers of patients suffering from these are clearly far, far fewer, but I would agree that there is an unmet need there. And if you go and talk to clinicians, they all agreed on pretty much 3 things that is needed in GI. They are desperate for a prokinetic, absolutely desperate. The FDA is desperate for a prokinetic. Cisapride was working, and it was withdrawn. Tegaserod, I have to say it was a good drug, it was our drug. It was okay. In those patients that responded, they responded very, very well, and that's been withdrawn. So the FDA has actually told the pharma companies, please give us another prokinetic. Then if you talk to clinicians and patients, the pain associated with irritable bowel syndrome remains an unmet need. We don't really know how to treat that. I'll come on to this later, but I think over the past few years there's been a bit of a movement in regarding IBS as a chronic pain syndrome. And that's probably not true, and I think that may have misled us, actually, especially in choosing the targets that we're going to work on to treat IBS. But finding a way of treating that, whether it's directly affecting the pain or whether it's affecting the motility, which we hope might indirectly affect the pain, that is an unmet need. And then in IBD, Crohn's disease and UC, I'd say that we're fortunate there are therapies. So the anti-TNFs work. Again, in those patients where they are effective, they work very well. And on the whole, quite safe. but again, we could argue that they haven't been around long enough to understand the long-term safety. What we need in IBD is to be able to maintain patients in remission. So using steroids or using anti-TNFs, we can induce the remission. Now we need a safe therapy, ideally orally delivered, to keep them in remission. And then, as Karen mentioned, fibrosis, again, is a big need. And we can argue actually about where all these fit in this sort of map. But when we're choosing what to work on, and actually when I started my job, which was to start up a GI discovery unit, and I said, okay, what do you want me to work on? And my management said, whatever you want, you choose, which actually wasn't terribly helpful. But if you look at it this way, where's the unmet need? That's obviously the first starting point, is the unmet need. And it's not commercial value at all. Everybody thinks pharma is just out for money, and clearly they are. But it's not the commercial value that drives what we work on. It's actually what the patients need. Then, of course, there's no point trying to treat that if there's no way of accessing. So it has to be scientifically tractable. Now, where you put these in the boxes, I think we could all argue. Excuse me. Yep. Obviously, this is where we want to be. So we need to be in an area of high unmet need, and we need to understand the disease enough so that we know how we can try and tackle this. And I would argue that maybe of those diseases that I showed in the previous slide, gastroparesis at the moment is the only one that's up there in the top right. So there is a confirmed unmet need, and it's pretty straightforward of what we need to do. We need to increase the motility of the upper GI tract. And the easiest way, and all the work of Sanger has shown this, that if you stimulate these neurons, at some point we're going to stimulate the emptying of the stomach. IBD, as I said, I would say we understand quite well what's going on in IBD, especially with the recent publication of many genome-wide scans. It is giving us a lot more insight in what is going on. But we do have some drugs that are there, and this contrasts with the functional disorders like IBS, where I'd say we really have little idea of what's underlying these disorders after many years of research, but there's absolutely nothing now to treat them. So the unmet need there is undoubted. So what I'll do is I'm just going to choose a couple of these and discuss them in a little bit more detail of the challenges. So we'll start with IBS. It's close to our heart. In Novartis, it remains close to our heart, and we still think it needs treating. Huge prevalence of IBS. About six, the numbers vary, but we're in the so many millions that whether it's 60 million or 70 million doesn't really matter. It's absolutely huge. And IBS accounts for around half of all visits to gastroenterologists. So this isn't the GP, these are the clinics, the gastroenterology clinics. And it really does affect the quality of life. The problem is, in terms of treating it, is it's not a homogeneous disease, it's heterogeneous in terms of its symptoms and actually the pattern of problems that people present with. One thing is consistent, and that's the abdominal pain. About 80% of patients will report abdominal pain. And this isn't the sort of sharp, lancinating pain that chronic somatic pain conditions present with. This is a very diffuse pain. You can't really localize it in the abdomen. Of course, that's a defining feature of visceral pain, and it's associated with cramping frequently. So along with abdominal pain, for IBS, you have to have an altered bowel habit. This can be either an increase or a decrease, or rather confusingly, alternating between diarrhea and constipation. Genetics, is very little known about genetics of IBS. There haven't been any big array studies or scans done in IBS. There's been some candidate genes that have been identified, and SNPs in some sort of mediators have been shown in IBS patients. But whether we could really regard those as a marker of disease, I strongly doubt at this stage. We do know there are risk factors. Again, these are widely known. Infectious stress, especially early life trauma, certainly predisposed to the development of IBS. And the etiology, as Brendan says, is very vague, but there are some features of IBS, if you go looking, that are reasonably consistent. there is visceral hypersensitivity in a proportion of patients. The exact proportion really varies between studies. And I think earlier this decade, we all got very excited and said, okay, this is the key feature of IPS, and we can hang our hat on that. And numbers of like 80% of patients have visceral hypersensitivity were bandied around, and that's probably reducing now. Clearly, there's dysmotility, and inflammation actually is being confirmed as a feature of IBS. It's not the frank inflammation, of course, that you get with IBD, but there clearly is a low-grade inflammation in a proportion of patients, in particular those associated with infectious, so post-infectious IBS. And there's an intriguing story that maybe mast cells might be sort of pathologically involved with the disease. We're not sure because it hasn't been tested in the clinic yet. Therapeutic options, we've heard before, and it's a problem. So we had the prokinetics, tigacerod and cisapride, both of which have been withdrawn, and the 5-HT3 antagonist for IBSD, which was also withdrawn. Now, these drugs worked. They worked in a proportion of patients, but they worked enough, certainly, to make it worthwhile. And the safety risks, they're there. These are the numbers for tigacerod, 0.1% of patients treated with tigacerod in published clinical trials had an ischemic event. These patients all had a history of cardiovascular disease. And Lotronex, the proportion actually isn't any higher. Unfortunately, there were some deaths with Lotronex because of the complications with the severe constipation and the colitis. But I think it's interesting if you compare this with the non-steroidal risk, where we have huge numbers of people still dying through gut bleeding due to non-steroidals. The proportion, admittedly, is much, much less because the prescription of non-steroidals is so huge. Benzopride, unfortunately, has been withdrawn, probably didn't work well enough, not well enough for the biotech to proceed. So the only drug that we've got at the moment is lubiprostone, which is a chloride channel activator, epithelial chloride channel activator. It's essentially a laxative, and it does work a little bit, but it's really poor efficacy in the clinical trials. What we were looking at with the Lotronex and with Zelnorm was up near 15 to 20% efficacy in symptom relief above placebo. But now we're glad that we've got something that is giving 88%, and that's really not very good enough. So this is the challenge. We need something else. How we go about finding something else is difficult. And I keep saying this thing, we need something to hang our hat on, because we haven't got any genetics, because we haven't really got a defined pathology, an organic basis to the disease. But these are consistent features of IBS. Reduced pain threshold, as I mentioned, this has been relatively consistently shown in different sorts of exploratory studies. You probably can't see here, we've got the controls, this is IBS, so this is the rectal pain threshold. This is where you essentially insert a balloon into the colon and you dilate it, you increase the pressure, and then the patient reports when this feels painful. And the threshold of pressure for that for IBS is really reduced, and it's nothing to do with just the bowel movement, because it's not affected in functional constipation, and dyspepsia is also not different. So this does appear to be something that's specific for IBS. Clearly, there's changes in motility, up or down, depending if we've got diarrhea or constipation. And the permeability is really something that's been highlighted just in the last couple of years, and this could be quite exciting. So this is looking at essentially the sorts of studies that Karen was looking at in the previous talk. You can do this in patients where you just give sugars that are permeable at different parts as it progresses down the gut. And you can see in a large, about 50% of IBS patients in this study had an increase in epithelial permeability. Now, whether this is now going to be causing some of the underlying features, you could imagine cells, bacteria accessing the submucosal layer, which they wouldn't otherwise do. mediators from those could then obviously go on and sensitize neurons and perhaps underlie these features of the pain. But this is something that's just coming out. So what we don't know is which of these aspects we need to look at to choose our biological targets to treat the totality of the IBS symptoms. Will a visceral analgesic be enough? Will something that affects motility alone be enough? I think probably not. And then what we have to do, obviously, preclinically, is look at these in animal models. And these are, they're okay, but I don't think that you can claim that they're any kind of model of IBS. We've just taken those symptoms and individually are able to look at each one and see which of our targets or potential therapies may influence one of these features. So this is transit. It's a glorified charcoal meal model that's been around for many, many years. And here we're just giving a high molecular weight sugar, and you can track its progress down the gut and influence that with drugs that may speed up or slow down motility. It's quite useful. If we want to look at that on a more gross scale in animals, we've got this highly complex model of a rat where we just count the fecal pellets. It takes a lot of skill to do this, as you can imagine. And this is a water avoidance stress, where obviously stress will increase defecation, and something like 5-HT3 antagonist reduces that nicely. So we validated the model. And then the visceral hypersensitivity. You could argue that this is translatable because it's essentially what was done in man when I showed you that slide of the decreased pain thresholds in patients. So you put a balloon in the colon, you increase the pressure, and then we're looking at a sort of downstream reflex response to that increased pressure that we're interpreting as a response to pain or nociception. So it's just the twitching of the abdominal muscles. And this works quite nicely. What we then want to do is to produce a state of hyperalgesia or the increased pain that we think IBS patients have. And we can do that through stress. Here we've administered an inflammatory agent, TMBS, into the colon. And you can see this black bar is the control. If you inflame the colon, you get a greatly increased EMG response to your colorectal distension. That's what we call the hypersensitivity. And we can reduce that again with Alocitron, the 5-HT3 antagonist. And it isn't reduced, apparently, with diclofenac, just a non-steroidal. So we would say it's not just due to the inflammation. So again, we would say, okay, the animal model is nicely validated. We'll test lots of new targets or lots of new potential therapies through this and then go into man. Problem is, it hasn't really panned out very well. There have been some exploratory studies in man looking at this colorectal distension paradigm where things that work in the animal model haven't worked at all, notably NK3 antagonists. Other things have been a great success. Pregabalin, which I think is a terrific drug, actually, works really well in this, worked really well in man. So it's not entirely predictive yet. So then we go to the clinic. So we carefully set up these models that look at the particular symptoms, if you will, of IBS. Then we go, okay, we've got a target and we've got a compound hitting that target and it affects motility and it affects pain or it might affect permeability, and it's all very careful. Then we go to the clinic, and what we do is we ask the patient how they feel. It's just not related at all to what we're doing preclinically, or preclinically is not related to what we are able to do clinically at the moment. So this just shows one of the published studies with tegaserod. This is Zelnorm. And you can see that there's a separation from placebo of around 15%. So it's working in these IBS patients with constipation. Still, in all of the IBS trials, and this is what's been asked for by the regulatory authorities, are PRO, primary endpoints. That's patient-reported outcomes. Essentially, it's asking them what they feel. These are either binary questionnaires, so in the last week, have you had adequate or satisfactory relief of your symptoms? Yes or no, that's it. Or we are now incorporating them more into a global symptom score. So we've got this IBS symptom severity scale, where on the questionnaire, it has measures of pain, bloating, bowel dysfunction, and quality of life. But again, it's just asking the patient how they feel. And we score it, and if you're below 75, you're normal. So hopefully most people here will be below 75. And if you're above 300, then you've got severe IBS. And the problem with any sort of PRO, patient-reported outcome, endpoint in clinical trials is that you will get high placebo rates, and you will need high numbers of patients to get any kind of statistical significance in your measure. And I've put over 100. I think if we were to use this in a phase 2A trial now, we would need at least 100 patients per group. And actually the Alosetron and Tegaserod studies all had several hundred patients per group, not even in the whole trial. So this, I think, is a very profound problem with IBS, is exactly what are we going to measure in the clinic. And we still don't know which of those pharmacodynamic endpoints that we do measure preclinically, we don't know which one of those being affected will translate into an overall positive effect on these disease scores. I think it can be different. I think this is quite a nice story. Linaclotide is a new drug that is coming through to treat IBS. It's acting on another chloride channel, the CFTR channel, which is on the apical surface of the gut. And actually it doesn't act on that channel directly. It acts on the membrane-bound guanylate cyclase, increases cyclo-GMP in the cell, which activates CFTR, which leads to chloride flux into the lumen, which is followed by water. So it's a mechanism that is pro-secretory. So it would work in principle on anything associated with constipation, so IBS-C. And these are studies just from animal models, just a quick experiment we did, looking at this FIT-C transit down the gut. And you can see with linaclotide, it's increased the front of the movement excuse me, of this FIT-C. So it works on increasing motility in animals. This is a different visceral pain model where we've, I think it's mustard oil into the colon, and we're looking at the referred allodynia on the flank. And again, linaclotide is very effective in reversing this visceral pain, nearly as good as Alosetron. Now, clearly with this sort of mechanism, you would predict the increased transit. The reversal, apparently, of the visceral pain was somewhat surprising, but others, including Microbia, who developed this drug, have reported that. We've found, actually, GCC is not just here. We've found it in DRGs as well, sensory neurons as well. So maybe there is some kind of effect there, although it's a peptide and it's completely non-absorbed. So it's quite intriguing, actually, as how this drug works. What I think is cool is that they show that you don't have to go into these large trials with a patient-reported outcome. So in the first trial, which used to be called phase 2A and we're now calling proof of concept, it's a small trial, only had 12 patients per group. They were just looking at motility with scintigraphy, which is a kind of radio-labeled method of measuring motility, and they showed an increase in colonic transit in IBS patients. And this actually was also translated with the pro-secretory effect into an improvement in stool consistency, which is absolutely what you need with IBS-C patients. So that was good, significant increase in transit time, nice paper, published, do your next trial, the phase 2B, and now you work up into the 400, really confident that it's worth investing more in this drug because it is doing something that you would predict from its mechanism and that you would also predict would have a benefit in IBS patients. And again, they got really stonking efficacy, actually, over placebo, and the phase 3 trials are ongoing. So it's just an example of where we don't have to use the PROs. We will ultimately have to do it unless the regulatory authorities completely change their mind, which is unlikely. But we can use, if you will, a kind of surrogate readout of efficacy of these new targets. Now move on to IBD, which is really the other end of the spectrum of GI disorders. In terms of numbers, it's completely different to IBS. There's about, I have a number here of 1.5 million in the US, maybe up to 2 million worldwide. It's largely restricted to the developed world, and the symptoms are different. You know, they're really quite dramatic and very distressing symptoms in these patients. The contrast with the functional disorders is the genetics. So now, with the genome-wide scans that have been looked at and the meta-analyses, there are multiple SNPs identified, and there's not just a list of SNPs, they're actually bracketing themselves into types of mechanisms. So there's some around the adaptive immune system, there's some around the barrier, the epithelial barrier function, and there's some clearly around recognition of the bacteria, the commensal bacteria in the gut. So this could help us, actually, not only in identifying the mechanisms or the individual targets to look at, but hopefully down the line in picking the patients that we should enter into our trial. Pathology you've seen in the previous talk, and really the mechanisms we know much more about IBD, and it's pretty well accepted now that it results from a loss of tolerance to the commensal bacteria in our gut. For some reason, we start reacting against these. And this is particularly the case when you put it on an environmental and a genetic background. There are some patients that are susceptible. Now, I think there's a bigger problem, in a sense, with IBD in terms of preclinical work and our animal models. We've got a huge number of animal models that we can use if we want to look at targets that we will say will be potential to treat IBD. DSS, I'll come on to in a minute, is the most widely used. Essentially, this is a chemical that really damages the colonic epithelium, and that the permeability of the epithelium really increases. Bacteria then cross over and establish this innate and then adaptive immune response. Then we have models where we use haptens, TMBS or oxazolone. We put them in the colon. These are more chronic models lasting a couple of weeks or more. And they again lead to this sort of ongoing inflammation. And we've persuaded ourselves really that the cytokine pattern with TNBS that we see is a Th1 type pattern, and that with oxazolone is a Th2 type. And then we take the even bigger leap and say, oh, well, this is a model of Crohn's and this is a model of ulcerative colitis. I think that's quite dangerous. IL-10 knockout actually could be a lot better, only because when it was set up, we didn't know this, but we now know that there are a number of SNPs in the IL-10 gene associated with ulcerative colitis in particular. But really, this has an ongoing epithelial barrier dysfunction. This model lasts many, many weeks, and it's the only model where we've got a spontaneous colitis developing. Difficult to use because every animal develops a colitis at a different point, but nevertheless, you could argue that this is the most disease relevant. Then there are others. T-cell transfer is looking at the adaptive immunity. We think that chronic TMBS has some features of fibrosis. All of these have disease, I say disease, have inflammation, signs of inflammation only in the colon. Crohn's disease, of course, is not a colonic disease. It can happen anywhere up and down the GI tract. So that's a problem. And we've got some models which are not in wide use yet that appear to have an ileal inflammation, and so hopefully could model Crohn's a bit more. Now, the most widely used of these is DSS, dextran sulfate, and you give this to a mouse in its drinking water for a few days, and what you have is a dramatic decrease in body weight that then recovers. And we have various scores that we can look at that together as a totality we call disease scores. So there's fecal scores in, whether there's bloody diarrhea or loose stools or no change. There's a decrease in length of the colon, don't really know why, but this is a marker of the disorder. And then the various cytokines go up. The ones that you would want to go up, that's okay. And you can see there's a profound inflammation. There's a real decrease in the architecture of the mucosa and an infiltration of inflammatory cells. And again, we can score that. This is a histology score, and all of these are reduced to varying degrees with a steroid. Other clinically used drugs, anti-TNF-alphas, don't really work in this model very well. 5-ASAs don't really work in this model very well. Anti-IL-12 works a bit in some labs, but not another. But nevertheless, we're fully confident this is the model of inflammatory bowel disease. My problem with it, okay, I'm just going to make sure I've got the next slide here. This is what I keep saying to people in my group, and they get really, really fed up with me, but what are we trying to treat? I keep saying we're not trying to treat a mouse, we need to treat men. And I found this quite horrifying. I just did a quick Medline search just before coming here, and in the past 3.5 years, there have been 400 papers published with the DSS model. About 250 of them claim a new target or therapy for IBD. And this struck me as quite significant. These are just the titles of some of the papers. We've got a therapeutic approach to experimental colitis. We've got efficacy in treating experimental colitis, efficacy in the model of this. Our focus has shifted. We're happy if we can treat the mouse. And I think that's something that we should bear in mind. And these are just some of the targets that have been claimed in the past 4 years to treat IBD based on this model. And call me a cynic, but I really don't think all of these are going to be good targets. And in fact, I'm not sure we need that many targets. But what it provides us with is this minefield that we've got to go through and somehow choose which one are we going to work on. Now, translating to the clinic, it doesn't really get much better, actually. What we use is essentially another patient-reported outcome. Crohn's disease activity index is a similar one for ulcerative colitis, the Mayo Clinic index, and it's just a series of features which are then weighted, and overall we get a score. And if you're below 150, I think you're in remission. If you're above 400, you've got severe disease. But again, they're pretty vague. Liquid or soft stools, a rating, how bad is your pain, a rating, how are you feeling today. And these are just 2 very recent trials. This is the pegylated anti-TNF-alpha. This is, I think, anti-IL-12. And you can see that even for this disorder, we've got a really defined pathology. We've got high placebo rates in the clinical trials. It's actually not any better than with IBS. We're up at 30 to 40%. And significance is going to be difficult to get a significant effect over placebo with those sorts of numbers. So I think this is my second to last slide. The issues with clinical trials in IBD are quite similar to IBS, actually, which surprised me. So for a typical phase 2A, we still need large numbers of patients. We're going to need at least 50 patients per group. We're going to need at least 12 weeks in a trial to see if we've got efficacy. Biggest problem here is recruitment. If you go to a typical site, they'll say they can give at best 0.3 patients per site per month. So that's about 4 new patients per year that can be entered into our trial to test our new drug. So you can see with that and the numbers required, you're going to have something like 50 sites participating in a trial, which is okay if you're using a standardized score. If you want to use something new, like a new biomarker or a new methodology, you're never going to be able to standardize that across 50 sites. And then there's the huge number of trials ongoing. If you go to clinicaltrials.gov, there's over 150 trials ongoing in IBD. There's just not enough patients around and new patients around to support that number of trials. Not all of them are testing a new drug, but over 100 of them are testing drugs. Sadly, there's hardly any that are testing new drugs. It's looking at different features of the existing ones. So we've got the high placebo, we've got a low sensitivity of this Crohn's disease activity index. And what we're not doing, we still have no way of looking and testing for the key unmet need in IBD, which is fibrosis, preventing or reversing the onset of fibrosis and maintenance of remission. Despite that, there's a lot of trials ongoing, but I would say there's little innovation. They're all around immunosuppression. There's lots of biologicals, which is exciting, but also, as you'd expect with this sort of disorder, but there's nothing much different here. Lots of anti-TNFs. The immunosuppressants. So finally, this is my last slide. I think we can agree from all the talks this morning that there remains a huge unmet need in GI diseases, and I think we can funnel down and choose those particular diseases that we still need to treat. It is a difficult area of drug discovery. I think I would argue there's a heavy reliance on unpredictive animal models, and we have to add something else to our level of knowledge to choose which targets to pursue, and in particular what we're going to look at in patients, so that we don't have to rely on these patient-reported outcomes. In some of the diseases like IBS, the functional disorders, we're beginning to understand more about them a little bit. We're creeping up in our level of knowledge of the underlying pathology, perhaps, but we still don't know which one of those features, if we affect it, or which combination will lead to and affect the overall disease. There's very, very little use of biomarkers. I know that's a little bit of a buzzword, but we absolutely do need that, because what we need to know is what are we going to measure in which patients, and we have to be able to choose those patients based on something in order to test our new drugs. Thank you.